The 10th and most terrifying plague of Egypt, the death of the firstborn. An event so selective it defies our understanding and remains one of history's and science's most fascinating mysteries. Was it divine intervention, an ancient punishment with unprecedented surgical precision? Or can modern science, equipped with knowledge in epidemiology, genetics, and toxicology, offer an explanation that resonates with natural phenomena? Join us as we delve into this investigation, seeking to decipher the enigma behind a plague that struck only the firstborn, questioning if it was, in fact, a tailor-made plague. The narrative of Passover and the final Egyptian plague, as described in the Book of Exodus, is one of the foundational pillars of Judeo-Christian faith and one of the most dramatic accounts from antiquity. To understand the uniqueness of the death of the firstborn, it's essential to contextualize it within the divine sequence of judgments upon Egypt. God, through Moses, initiated a series of 10 plagues with the unequivocal aim of demonstrating his power and compelling Pharaoh to free the Hebrew people from slavery. The preceding plagues, the turning of water into blood, the infestation of frogs, lice, flies, the livestock plague, boils, hail, and fire, locusts, and dense darkness, escalated in intensity and devastation, but they affected Egypt more generally. Each was a direct blow not only to Egypt's infrastructure and economy, but also to the gods they worshipped, challenging the sovereignty of Pharaoh, who was considered a living god. However, the 10th plague, detailed in Exodus 12, transcends the previous ones in its precision and psychological impact. The Bible recounts that at midnight, the Lord struck down all the firstborn in Egypt, from the firstborn of Pharaoh, who sat on his throne, to the firstborn of the prisoner in the dungeon, and the firstborn of all the livestock. The specificity of this plague is astounding. It did not strike randomly, but targeted a very particular demographic, the firstborn of humans and animals. The only exception was the homes of the Israelites, who followed divine instructions, sacrificing a lamb and marking their doorposts and lintels with its blood. For the Israelites, this ritual Passover would become an enduring memorial of liberation and divine protection. The death of the firstborn was not only the culmination of the plagues, but the final catalyst for liberation. Mourning and terror spread throughout Egypt. Pharaoh, hardened until then, finally relented, begging Moses and Aaron to depart with their people. This event not only marked the Hebrews' exodus from Egypt, but also established Passover as a central feast in the Jewish religious calendar, symbolizing redemption and the passage from slavery to freedom. The magnitude and precision of this plague raise profound questions that serve as the starting point for our analysis. Why the firstborn? This selectivity, so unusual in natural disasters or known epidemics, is the core of the mystery we seek to explore. The quest for a rational explanation for the selectivity of the death of the firstborn has intrigued scientists and researchers for centuries. While the biblical text directly attributes the event to divine intervention, science, in its inquisitive nature, seeks patterns and causes in natural phenomena that might have manifested in an extraordinarily specific way. One of the most intriguing and studied hypotheses in this context involves the possibility of environmental toxins or diseases with selective infection and lethality patterns. Let's consider the conditions of ancient Egypt. Life revolved around the Nile River and its annual floods, which, while vital for agriculture, also created an environment conducive to the proliferation of microorganisms. A plausible theory suggests that previous plagues could have created a cascade effect that culminated in the 10th plague. For example, the transformation of water into blood could have poisoned the aquatic ecosystem. The death of fish and the proliferation of amphibians and insects could have contributed to a significant increase in pathogens in the environment. The livestock plague, in turn, would have weakened the food and sanitary systems. In this scenario of ecological imbalance, food contamination would be a high risk. One of the most articulated theories proposes that the plague of the firstborn may have been caused by a mycotoxin, specifically aflatoxin, produced by fungi such as Aspergillus flavus and Aspergillus parasiticus. These fungi thrive in conditions of high humidity and heat, environments common after the Nile floods, especially in improperly stored grains. Aflatoxins are hepatotoxic and carcinogenic, but their more acute action can cause Rayleigh syndrome in children, a condition affecting the liver and brain, leading to encephalopathy and coma. The selectivity for the firstborn could be explained by consumption patterns. In many ancient cultures, including Egyptian, the male firstborn held a superior social and religious status. Consequently, they might have had privileged access to certain portions of food, perhaps the first grains from the harvest or older stored foods, which would be more prone to fungal contamination due to storage time and inadequate conditions. Additionally, the diet of firstborns might have included specific foods that, for some reason, were more contaminated or more susceptible to the proliferation of these toxins. In a society where food storage quality was precarious and knowledge about toxins was non-existent, continuous ingestion of small doses or a single large dose could have devastating effects, especially in children and young adults, whose hepatic and immune systems were still developing or overburdened. Another line of research speculates on selective infectious diseases. While most epidemics tend to be more generalized, some diseases affect specific groups with greater severity. For example, a bacterial or viral infection that manifested more virulently in children or in individuals with somehow compromised immune systems. Whooping cough or certain forms of bacterial meningitis, for instance, are particularly dangerous for young children. If the passing of the angel of death was a rapid and lethal outbreak of such a disease, the vulnerability of the youngest, who in many families would be the youngest firstborns or the only children, could have contributed to the perception of a selective plague. Science, in exploring these avenues, does not seek to discredit the biblical account, but rather to offer additional perspectives on how an event of such magnitude could have manifested in the natural and social context of ancient Egypt. The selectivity of the plague of the firstborn leads us to an area of science that explores individual and population predisposition to certain conditions: genetics. Could there have been an underlying biological explanation, a genetic factor that made the firstborn more susceptible to the plague? Although ancient Egypt had no knowledge of genetics, modern science allows us to speculate how certain hereditary traits could have created specific vulnerability. One of the most debated theories, albeit with limitations, is that of hemolytic disease of the newborn, also known as erythroblastus fetalis. This condition occurs when there is an Rh factor incompatibility between the mother and the fetus. If a mother is Rh negative and the father is Rh positive, and the fetus inherits the Rh positive trait from the father, the mother's body can develop antibodies against the baby's red blood cells. During the first pregnancy, sensitization is less likely to cause severe damage. However, in subsequent pregnancies, the presence of these maternal antibodies can lead to the destruction of fetal red blood cells, resulting in severe anemia, jaundice, and in severe cases, death of the newborn or infant in the first few days of life. The argument in favor of this theory is that firstborn children would be more likely to survive the initial exposure without major complications, while subsequent children might suffer more severely. However, this theory has a significant limitation. It would only explain the death of firstborns as newborns, not encompassing firstborns of all ages, including Pharaoh's son, who was likely a young adult, as the biblical account suggests. Therefore, while interesting, HDN does not offer a comprehensive explanation for all reported cases. Another line of genetic speculation considers the possibility of rare hereditary conditions that manifested more severely or lethally in firstborns due to environmental, nutritional, or developmental factors. In populations with a considerable degree of endogamy, the probability of expressing recessive genes for rare genetic diseases increases. If there were a recessive genetic disease whose manifestation was triggered by a specific environmental factor, and which was more lethal to individuals at certain developmental stages, this could create a pattern of selective mortality. For example, an enzyme deficiency that made certain substances common in the diet at the time toxic to these individuals. Furthermore, genetics can influence an individual's immune response to pathogens. Genetic differences can make some people more susceptible to infections or more prone to developing severe forms of certain diseases. If the firstborn, for some as yet unknown genetic reason, had a predisposition to a deficient immune response to a specific pathogen circulating in Egypt at that time, this could have increased their vulnerability. It's important to note that these are speculative theories and direct evidence is scarce. However, the genetic perspective allows us to go beyond purely environmental explanations, suggesting that the tailor-made plague could have found fertile biological ground in a specific group of the population, making them more vulnerable targets. This approach does not replace the historical or religious narrative, but complements it, offering avenues for understanding how biological phenomena can manifest in surprisingly selective ways. To unravel the selectivity of the plague of the firstborn, it is essential to delve into the intricacies of diet, food storage methods, and the environment of ancient Egypt. Egyptian civilization was intrinsically linked to the Nile River, which, with its annual floods, not only fertilized the land for agriculture, but also dictated the rhythm of life and, potentially, the health of its population. The Egyptian diet, at its base, consisted of cereals like wheat and barley, used to make bread and beer, which were staple foods. In addition, they consumed legumes, fruits, vegetables, and, for those with more resources, meat and fish. The way these foods were produced, processed, and, crucially, stored is a central point of our analysis. In an era without refrigeration or advanced preservation methods, dependence on granaries and storage facilities made food vulnerable to spoilage and contamination. One of the greatest risks associated with grain storage in hot and humid climates, like that of Egypt, is the proliferation of mycotoxin-producing fungi. We've already mentioned aflatoxin, but other fungi, like Claviceps purpurea, which causes ergotism, are also a possibility. Ergotism, historically associated with outbreaks in the Middle Ages, results from the ingestion of grains contaminated with sclerotia of the fungus, which contain toxic alkaloids. The symptoms of ergotism can range from convulsions and hallucinations to gangrene and death, and could easily have been interpreted as a supernatural affliction in an era without scientific medical knowledge. The hypothesis linking the plague of the firstborn to food contamination gained strength when considering the social status of the firstborn. In many ancient societies, the male firstborn enjoyed a privileged status, being the primary heir and often the first to be served at the table. This could mean they consumed the first portions of newly stored harvest or, paradoxically, the oldest portions of grains, which would be more prone to fungal contamination due to storage time and inadequate conditions. It's possible that firstborns, especially in wealthier families, had a slightly different or more abundant diet, exposing them to a higher load of toxins if the food was contaminated. The best portion or the first portion could, without the knowledge of the time, have been precisely the most dangerous. Beyond diet, water quality was a critical factor. The waters of the Nile, although the source of life, were also a vector for disease. Previous plagues, such as the turning of water into blood, would have severely compromised the drinking water supply. If the firstborn had access to specific water sources or were more prone to consuming larger quantities of water due to age or status factors, exposure to waterborne pathogens could have been higher for them. The complexity of the interaction between the environment, culture, and biology allows us to visualize a scenario where a natural event, albeit extraordinary, could have manifested with the selectivity described in the biblical account. This analysis does not dismiss the miraculous dimension of the event for believers, but offers a way to explore the possible physical manifestations of an event of epic proportions. The idea of a tailor-made plague, while rooted in an ancient biblical account, finds disturbing and fascinating echoes in modern discussions about science, technology, and ethics. The exponential advancements in biotechnology, genetics, and our understanding of pathogens allow us to contemplate scenarios that once seemed like science fiction, raising questions about humanity's capacity to create or confront threats of previously unimaginable specificity. From a public health perspective, the COVID-19 pandemic, with its global scale and differentiated impact on various demographics, serves as a recent and vivid example of how biological factors and social factors can create specific vulnerabilities within a population. Although COVID-19 was not selective to firstborns, it demonstrated how a pathogen can interact complexly with human heterogeneity, resulting in patterns of disease and mortality that are not uniform. Ongoing research into viral variants and individual immune responses only reinforces the idea that biology can indeed operate in a tailor-made fashion, even if without deliberate targeting. Furthermore, the field of rare disease research and personalized genetics illustrates how we increasingly understand biological specificities. Genetic diseases that affect only a few families or specific ethnic groups are tailor-made by the very nature of their genetic basis. Precision medicine, which seeks to adapt treatments based on an individual patient's genetic profile, is a positive example of this biological specificity. However, the discussion about a tailor-made plague also leads us to a darker territory, the concept of targeted biological weapons. The possibility, though highly controversial and subject to strict international prohibitions, of developing pathogenic agents that could exploit specific genetic or immunological vulnerabilities in certain population groups has been a subject of concern and science fiction. This raises profound ethical questions about the misuse of scientific knowledge and humanity's responsibility to protect life. The idea of creating a disease that would, for example, affect only people with a certain genetic characteristic, hypothetically, one that was more common in firstborns, if such a characteristic existed and were linked to their primogeniture, is an extreme and dystopian manifestation of the tailor-made plague concept. Reflecting on the death of the firstborn in the light of modern science is not merely a historical or religious exercise. It invites us to ponder the complexity of life, the interaction between biology and environment, and the ethical responsibilities that accompany our growing power to manipulate and understand biological systems. What was once explained solely as an act of a higher power now compels us to explore the limits of what is biologically possible and to question the very boundaries of our capacity for understanding and control. The tailor-made plague thus becomes a catalyst for crucial debates about the future of biotechnology and the nature of human vulnerability. Our journey through the enigmatic plague of the firstborn has taken us along complex paths, intertwining ancient biblical accounts with the most advanced scientific speculations. We've analyzed the possibility of environmental toxins, explored intricate genetic connections, and delved into the nuances of diet and environment in ancient Egypt. Yet, despite all theories and advancements in human knowledge, the veil of mystery surrounding the death of the firstborn remains. The selective precision of this event, which struck such a specific part of the population, the firstborn, continues to defy a single, definitive, and entirely satisfactory explanation, whether purely scientific or theological. It is at this intersection between the known and the unknown that we encounter the limits of human understanding. There are phenomena that, by their very nature, seem to transcend the capacity of reason and science to be fully unraveled. The plague of the firstborn, for many, remains an unshakable testament to divine intervention, An unquestionable sign of a power that operates beyond the natural laws our science is capable of deciphering. For believers, the plague's selectivity is proof of divine sovereignty and purpose, a miracle that by definition escapes empirical logic. For the scientific community, on the other hand, the plague event is a persistent historical and biological enigma. It is a continuous invitation to research, to search for a missing link, a combination of environmental, social, and biological factors that, under extreme conditions, could have culminated in an event of such specificity. The lack of direct archaeological data on the selective mortality of firstborns in the Egyptian population of that era makes empirical confirmation of scientific hypotheses difficult, keeping the event in the realm of informed speculation. Regardless of our personal inclination, faith, or science, the central theme of the death of the firstborn compels us to confront the vastness of the unknown. It reminds us that the universe is vast and complex, and not everything can be easily categorized, measured, or replicated in a laboratory. There are aspects of existence, both in the past and in the present, that challenge our taxonomies, our models, and our certainties. The plague of the firstborn is not just an ancient biblical account or an intriguing scientific case study. It is an eloquent reminder of our own intellectual humility. It highlights that, despite all our progress and knowledge, there are still deep mysteries that may forever be beyond our full comprehension. A perpetual invitation to reflect on what truly transcends our understanding, keeping the flame of investigation and speculation alive.